In this video, we'll be talking about epithelial tissue, the subclassifications of this tissue type. So let's start by thinking about where epithelium is and some of the basic characteristics. Is that epithelia are sheets of closely adhering cells, and they can be one layer thick or I can have multiple layers of thickness. Where do we find them? Well, they cover body surfaces like your skin, or they line body cavities or tubes. So really, epithelial faces either the outside world or faces into a space. And the upper surface is usually exposed to the environment or the internal space in the body. Often we have glands associated with them, and here's one of the key characteristics is that they are avascular, so that it does not contain blood vessels itself. And that may seem odd that these are living cells, but typically epithelial tissue is paired with other types of tissues that are vascular, such as connective tissue. And we can see in the diagram down here, just sort of point out where it is, that this is some tissue right here. And epithelial is either on the surface facing a space or the outside world, or it could also be, we could have cavities, blood vessels, things like that, and epithelial we'll find there. So when I'm going to look for epithelium or epithelial tissue, I'm not looking in here necessarily. I'm going to look for spaces and see if that happens to be the type of tissue that lines it. And as I said, the epithelium itself is avascular, but typically we do have a tissue that underlies it, that is vascular itself, and that'll allow for some diffusion of nutrients and wastes. And typically it's connective tissue that underlies it. Now, what are the functions? Well, the functions are it protects deeper tissue from injury and infection, produces and releases chemical secretions, excretes wastes, absorbs chemicals including nutrients, it selectively filters substances, and also it's often tied in with detecting sensory stimuli. Now, when I look at epithelial tissue, the cells are held very closely together. When we deal with histology, we're often looking at the ratio of cells to matrix. So in epithelial tissue, the cells up here, you can see the cells basically adjoin each other. And I don't have much material, if any, between the cells. And the material between the cells is what we refer to as matrix. Now we're going to see down the road that some tissues have very sparse cells, yet they have a lot of matrix around it. This is not the case with epithelium or epithelial tissue. It also has a high rate of mitosis. And not surprisingly, because if you're a cell that lines a cavity or face the outside world, your outer cells of your layer are likely to get sloughed off or damaged. So you're going to have to replace them on a constant basis. So there's a lot of mitosis that we can see. As I said, they're packed closely together. Also, here's a feature, the basement membrane. And the basement membrane is that little strip that basically separates or attaches, maybe is a better way to think of it, attaches the epithelium to the underlying tissue. So it's a layer between the epithelium and the underlying connective tissue. What's in here? It's often a mixture of collagen, reticular fibers, proteins, glycoproteins, and other types of protein carbohydrate complexes. And often it almost acts, you almost have these little molecules, almost like cell adhesion molecules that help to hold this in place, almost like a Velcro will hold it in there. And it helps to anchor the epithelium to the connective tissue below. Now, whenever I look at an epithelial tissue, it could either be one layer thick or it could be multiple layers thick. Now, when we talk about the basilar or the basal surface, that is the bottom surface. So these would be the basilar cells. The apical surface Apical surface would be the cells that face the outside world or face a cavity. Now, not surprising, the lateral surface is the lateral surface. Now let's start looking at sort of some definitions of how we describe the subtypes of epithelial tissue. If I say simple, simple means it's one layer thick. And then what I do is I identify it as being simple, and then I'm going to use a descriptor to say what shape it is. And that's how epithelial gets its name. It's based on, the first name is based on how many layers, the second name based on its shape. Now in simple epithelial, it's one layer thick, it's named for the shape of the cells, and all the cells touch the basement membrane. In stratified, stratified means layers. So here's stratified. You can see that there's a number of layers. So it contains more than one layer. Now how do I name a stratified epithelium? Because remember, it's how many layers, so I already know it's stratified, which means a number of layers, but then I have to describe the cell shape. But here's the problem with epithelium. Often the cell shape is different at the basilar versus the apical side. Well, what we do is we will name it after the shape of the apical cells. So the cells here, the cells that face either the outside world or inside a cavity. Not down here. We don't base it on those names. It's the upper ones. So these tend to be very flat cells, and we're going to see shortly that is called squamous. So this would be a stratified squamous type of epithelium. So here's the names we get to use when it comes to epithelium. We have simple. Right? Simple means one layer. By the way, there's a basement membrane. We have stratified. Stratified, basement membrane once again, and it's a number of layers, but you see how the cell shapes change? And remember, we're going to name it on the outer apical surface cells, not the ones down here. And then we have pseudo-stratified. Remember, pseudo means fake. And if you had a quick look, you'd see nuclei at different heights, and you may think, oh, that looks like it's a number of layers. But technically, this is not a stratified tissue because all the cells actually touch the basement membrane. So we call it pseudo-stratified. Now, how do we describe the shapes? If the apical cells are flattened, we call them squamous. If they're cube-like, we call them cuboidal. If they're shaped like a column, we call them columnar. So we have simple or stratified or pseudo-stratified as the layer names. And then when we add the shape of the cell, it's either squamous, cuboidal, or columnar. Now, simple epithelium, we can have simple squamous, simple cuboidal, simple columnar. Remember, it's only one layer thick. We also have pseudo-stratified. And as I said, it's called pseudo because it looks stratified, but it really isn't. Now, there's a group of cells that we will see in some of these tissues called goblet cells. And goblet cells are very important. They look like a wine glass shape, and they secrete mucus. We tend to see these in simple columnar and pseudo-stratified epithelium, and we'll see some examples of them. So let's have a quick look. Now, this is what we'd be faced with if we're looking down a microscope. This is an artist's diagram, so let's start here. Now, this is taken from an organ, and organs are made of two or more tissues, so we got to identify where we want to be. This is not what we're looking at. That is actually a different type of tissue. But here is what we want to see. Now, how do I know what I should be looking for? Well, I should have sort of a preconceived notion. I should have a checklist that if I'm looking for epithelial, it should face the outside world or into a cavity, and I should have an idea what the basic shapes are. So here we can see that this is one cell layer thick, and you can see that the cells are fairly flat in shape. Very flat in shape. So, not surprisingly, what we're looking at here is simple squamous, and you can see it here on a photomicrograph. The last thing we looked at was an artist's diagram, and you can see the cells are quite flattened, etc. Now remember, that underneath here I would have a basement membrane, and that would connect it to the underlying tissue. So I'm looking at simple squamous. It's a single row of cells. Now, this is really good for rapid diffusion or transport of substances or secretion of fluids. I wouldn't ask you at this point to memorize where they are. We'll see them as we come, but just as an FYI, the alveoli, glomeruli, endothelium, serosa, these are all areas where you have to exchange a lot of materials. Let's go to simple cuboidal. Now the name tells you everything you need to know, right? Is that first I'm going to look into, it's going to face spaces. That's one of the things about epithelium. Next thing is, I look at it, it's only one cell layer thick. And you can see the shape of the cells. They're about the same height as they are wide. So those are a cuboidal type cell. And we can go to the actual photomicrograph, and you can see here's a space, here's a space, here's a space, and in this tissue I can see there are the cells, and yeah, they are a simple cuboidal type of cell. So it's a single layer of round or square. They're good at absorbing, secreting, mucus production, movement, and there's a number of organs where they exist. Now let's go to the simple columnar. Remember, columnar means column-like. And let's look at the artist's diagram. We can see here that the cells are very tall versus how wide they are. Even look at the nuclei. The nuclei become elongated also. Very prominent feature, elongated nuclei. Now here's the goblet cells. These are the mucus-producing cells. You can see them right there. This one, and they lead out to the surface. This one, you don't see it leading to the surface. It happens to be that the plane of section didn't show the rest of it heading up there. But if you had looked at a histological section just before this or just after, you would most likely see it continuing up to the surface. So these are the goblet cells, the G for goblet cells, okay? And of course, we have a basement membrane down here. Let's look at the photomicrograph. Once again, you can see these very elongated, tall nuclei. You can see that the cells are columnar, they're column in shape, and you can see here are the goblet cells. Goblet cells appear very clear, fairly clear compared to the surrounding cells. And in this case, we also have microvilli. And you can see that they're so dense in numbers that it looks like a brush. So often they're called brush border cells. And where do we find these? Well, we find them in places like the GI, the uterus, the kidney, uterine tubes. The goblet cells produce the mucus. We have the oval nuclei, and they're tall and narrow cells. And often we see them where we have absorption or secretion, and not surprisingly, because we often see the brush border or the microvilli as part of them. Now let's look at pseudostratified. Pseudostratified, now remember from the definition that they all touch the basement membrane. Now it may not be apparent in this picture that they all touch it, because of course some of these cells, the bottom bit may be just out of the plane of section. So we can't quite see them, but we'll have to take it for granted that if you notice that most of them that we can see do touch the bottom. And in all fact, they will all touch the bottom. And here's the basement membrane down here. Not surprisingly, you should be able to recognize these as the goblet cells, right? And remember, they're the mucus-producing cell. Now in this case, we get to see another feature, is that we're not looking at microvilli, we're actually looking at cilia. And remember, cilia have that power stroke and recovery stroke. And not surprisingly, wherever we see, not at wherever, but often when we see the goblet cells, is that we often have cilia associated with them. And of course, here, I'll just show you one of the locations where this actually exists. This is in the respiratory tract. So think about the respiratory tract. I'm breathing in air that's full of little particles, and I don't want them to get down into the alveoli. So not surprisingly, you can see we're looking at sort of the upper respiratory tract right here. This is where it's from. And not surprisingly, what happens is that we basically put out layers of mucus, little bits of mucus out here. And the job of the mucus is to basically trap particles or trap bacteria. And the cilia, through their power stroke and recovery stroke, will basically push the mucus up and out. So that will move the mucus up and out, and often it just gets cleared down into the stomach, or it potentially could be coughed out also. And this is normal process. Now we can see when this doesn't work that anyone who's a smoker, long-term smoker, often what happens is that they've actually damaged their cilia here. So they're no longer able to effectively move the mucus out, especially at night. And what happens is that, of course, first thing in the morning, when they wake up, they have this mucus that's sitting there because they have immobilized cilia, or the cilia have been destroyed. And what they do is they mechanically remove the mucus by basically coughing. That will dislodge and force the mucus up and out. So those are pseudostratified. Now here we're looking at stratified. So when I'm talking about stratified, it's 2 to 20 layers. The deepest layers attach to the basement membrane, and there's 3 main types we're going to look at. And remember, we call the tissue by the shape of the apical cell. So we have stratified squamous, stratified cuboidal, and stratified columnar. There's not many examples of those. And then we have another one, which sometimes is called transitional, or you can call it urothelium. Why urothelium? Because it really shows up in the urinary tract, or parts of it. So the stratified epithelium, it's the most widespread. The deepest layers undergo continuous mitosis. They have to replace the apical cells. And the cells at the bottom basically divide and work their way up through the layers, and they transform in shape. We'll see a great example of this when we look at the skin next semester. And the outer layers could either be keratinized, and such as on the skin surface, and this makes it very abrasion resistant. They build up a lot of these protein complexes called keratin. Now, we often lose the outer layer, say especially on the skin, through exfoliation or desquamation, and that's just basically losing or sloughing out off the apical surface cells. So let's have a quick look. We're looking at keratinized, stratified squamous epithelium. And let's look at the artist diagram. Let me just outline where the basement membrane is here. So the basement membrane is right along here. And below it is a different tissue. So this is not the tissue we want to look at. What we want to look at is this tissue. And you can see that certainly it's more than 2 layers thick. It's multiple layers. This is actually thick skin, so it has a lot of layers. And you can see that the shape of the cells change significantly as we move up. And once I get to the outer layer, I'm really looking at flat, dead skin cells. These are skin cells in this instance. So we're going to name the tissue based on the outer layer. Let's go to the photomicrograph. Once again, we can see that this is the thickness of the tissue. We can see that the cells at the basal end look quite different. They multiply down here, they move their way up over time. The cells at the top get sloughed off, and they're replaced by cells moving up. And the cells here are very flattened and basically dead. So it's a keratinized, stratified squamous epithelium. Keratinized means that I've added proteins, like keratin proteins in there, to armor plate these dead cells to give more resistance. Let's look at non-keratinized. And non-keratinized, once again, it's a stratified squamous epithelium. So it's a number of layers thick. You can see that the cells down here are a different shape than the ones up here. Now, it's similar to the keratinized epithelium, but the surface layer are not dead cells like we saw with the keratinized one. And in this case, this is not the outside surface of your skin, but this does give some abrasion and penetration of pathogen protection. So it acts as a barrier. The tongue, the oral mucosa, the esophagus, and the vagina. These are parts of the world, parts of the body that potentially face the outside world. So it's good to have this barrier. Now, stratified cuboidal, we can see it. You can see that here it's a number of cell layers thick. So it ticks all the boxes for that. The cell shapes are more or less cube shaped. They're about the same height as they are wide. You can see this in the actual photomicrograph. You can see that there's multiple cell layers here. And it's 2 or more cell layers thick. The surface cells, remember we base it on the surface cells, are round or square. And what do these do? Well, we see these involved in sweat production, sperm production, ovarian hormones, and so we find them in sweat glands, ovarian follicles, and seminiferous tubules. And we'll see those structures in other courses. And the last one we're going to look at is the urothelium. It used to be called transitional epithelium. It still is in many books. And this is a multi-layered epithelium. And the surface cells actually change from round to flat when it's stretched. Because look where this is. Let's take the best example. It's in the bladder. So as the bladder fills, kind of like a balloon, it stretches this layer and it changes its shape. But you can see that once again, it's a number of layers, so it is stratified. You can see when the bladder is not distended or stretched, the cells are fairly round. But when it does distend, the cells will actually flatten out more, so it undergoes that transition. And that's the urothelium. So that's a quick walk through the epithelial cells, and we're going to leave it there, and we'll pick up with the connective tissue.